Size range. You don't have to know numbers. What it focuses on, microscopy. Okay. So light or electronic. Branches of microbiology. I ask you the type of microbes, you tell me which branch studies it. Does that make sense? Phycology, algae, mycology, fungi, and so on and so forth. Types of microbes. For each type that we discussed, it's a slide number 2. I even remember the number. For this, whether it's multicellular or unicellular, okay, or can be both, like algae and fungi. They can be either, okay. No cellularity in viruses. Be able, when I say can you describe each type in one sentence, you will not have to do that in multiple choice exam. But if I give you a description, you have to recognize it. Does that make sense? Okay. Like acellular organisms that consist of at least protein and nucleic acid. You see acellular, you see viruses. Got it? Okay, things like that. Bacteria or archaea look like. What I refer to is have a decent, it's gonna be repeated, okay, again. Have an understanding of what the cell looks like. There's membrane, most of them have wall, some of them have glycocalyx, cytoplasm is inside, there is no nucleus, there are no membrane-bound organelles, and so on and so forth. So it kind of gives you what are the main parts of the bacterial cell, what are their functions. Okay? Got it? Protozoa. Mandatory. What do they lack? What do they not have? Cell wall. They're eukaryotic. Structural features, that's pretty much lack of cell wall. How can they be classified? Remember those 4 categories? Sarcodina, Mastigophora, Celiophora, Epicomplexa. So those 4 things, why classifications like that? So Mastigophora flagellated, Celiophora ciliated, Sarcodina pseudopodia movement, and Epicomplexa do not have any movement appendages. Fungi, unicellular or multicellular. Yeasts are unicellular, molds are multicellular. Dimorphic fungi can be both. Have cell wall that is different from bacterial. That's it. Okay. Helminths, multicellular worms. 3 groups. So, okay, you have flat and round. And flat are tapeworms or cestodes, and flukes or trematodes. Okay, and round are nematodes. Know the names. Viruses, acellular organisms. Are they alive? No. What do they consist of? Nucleic acid, protein, capsid. For naked, nucleic acid, protein capsid, and envelope made of proteins and phospholipids for the enveloped ones. How are they classified? Based on the type of the genome. So have an idea what types of genome they have. Single or double strand DNA, single or double strand RNA. Does that make sense? So pretty much I'm answering those questions, but if you find yourself struggling with some of them, by tomorrow let me know. How ubiquitous microbes are? That's a stupid question everywhere. Highlight the importance of microbes in technology, nature, disease causing. So pretty much know which industrial applications microbes are important for. If I ask you this, I'm going to be very explicit. Look, if I ask you, if you have an answer about microbial importance in production of steel, if you ever saw how steel is produced, you know that there's no place for microbes. It's not even a place for humans. Okay? Or car making. I think microbes important in car making. No. Okay? I'm going to be very explicit. Same goes for diseases. Have an idea, like bone fractures, probably microbes are not terribly involved unless you drop the incubator on your leg. Taxonomy. Whitaker, 5 kingdoms. Know them. I'll ask them. What it is based on. Okay? Morphological evolution and morphological relationships mostly. Woe's folk system based on the genetic sequence of 16S. Ribosomal RNA, 3 domains. I'll ask you about 3 domains. I'll ask you which 2 are related more than another. Archaea and eukarya closer to each other than both of them to bacteria. We talked about that. Binomial nomenclature. Giardia lambli is the species of the genus Giardia. Does that make sense? You had this question before. Get ready for it. Hydrogen bond between oxygen, partially negative oxygen, partially positive hydrogen. Okay? What is pH? Acidity. If you go into acidic, it's low. If you go into the basic, it's high. Carbs made of monosaccharides, which are carbon, hydrogen, oxygen. When you dehydrate monosaccharides, they form polysaccharide. When you hydrolyze polysaccharide, they form monosaccharide back again. Remember, I have written that reaction. Okay, so review that, how they are formed. Function, energy structure. Lipids. 3 lipids that you have to know is triglycerides, phospholipids, and then that, sterols and hopanoids. Again, I'm not going to present you with a structure of hopanoid and you will have to tell me whether it's a hopanoid or it's a sterol. No. I will focus more on the function. For triglycerides and phospholipids, know the parts. Am I clear? Great. Yeah, you got it here. Proteins. What are they? Made of amino acids, 4 levels of structure. Now, if I will ask you, it's not just primary, secondary, tertiary, and quaternary. Understand what each level represents. Like primary, it's a sequence. Secondary structure is the beta sheets and alpha helices. Tertiary structure is the beta sheets and alpha helices folded together. Okay? When structure is destroyed, it's denaturation. Good? So that's pretty much what I refer to here. How the process of structural alteration is called. That's denaturation of the proteins. Nucleic acids made of nucleotides. What is the function of nucleic acid? Storage and transmission of genetic information. What's the difference between DNA and RNA? DNA is double-stranded, RNA is usually single-stranded. And understand the concept of complementarity. In the DNA, A is complementary to T, G, In RNA, A is complementary to U. In RNA, instead of T, you have U. AUGC in RNA, ATGC in DNA. So far, clear? Understand that what is genome? All genetic information of the organism. What is gene? A certain part of the genome that encodes for a specific protein. Trait can be encoded by one gene or multiple genes. Genotype, genetic information regarding organism. Genotype defines phenotype, the appearance. Gene expression. When genes are expressed, well, it's really hard to define what is gene expression. When genes are expressed, you get phenotype. Understand that not all the genes are expressed at the same time. Mutation is the change in the DNA, and it's an important step in evolution, right? Does that make sense? Type of the microscope that we use. Light bifocal binocular compound. Okay, understand those terms. Understand this. I pretty much have definition. Be able to calculate magnification. Okay. Multiply objective by eyepiece. Resolution, contrast. Limits of the resolution. Wavelength of light sets the limit for light microscopy resolution. Does that make sense? Who invented Lube and Hooke? And Hooke, two guys invented microscopy. Major parts of the microscope and their function. Remember that picture? Review it. Those questions are going to be very straightforward. This part of the microscope is where the specimen is placed. Mechanical stage, for God's sakes. Major types of light microscopy. I will do my best to be fairly simple and straightforward. You saw the questions on this quiz, right? Like this type of microscopy observes the specimen against dark non-lit background, dark field. Okay. Two types of electron microscopy that we talked about, transmission and scanning. Understand the difference. Yes? Say again. Understand the pictures? Yes. I'm not going to ask you about the pictures for the light microscopy. Absolutely not. Okay? Why do we fix the specimen? So it doesn't move. And we can stain it afterwards. Why do we stain the specimen? To increase the contrast. Types of the simple stains. People, you have the list on the table. You saw the questions on the quiz. It's going to be similar. Same goes for differential. This stain detects, obviously I'm not going to ask which stain detects capsule. I'm not going to ask which stain detects flagellum, right? It's just, the answer is in the question. So, but please know them. Please understand what they do. For steps of microbial cultivation, it's kind of counterintuitive. I have 5. Inoculation, what's next? Incubation, what's next? Isolation, and then inspection. I put inspection and identification in the same step. You're not going to get a question, how many steps are in the microbial cultivation? 1, 2, 3, 4, or 5? God forbid, no. Okay? But understand what we do during each step. Got it? Classification of media. Physical state and composition is simple. Purpose, we have general, enriched, selective, differential. Know what they are. Understand the term fastidious. Fastidious belongs to bacteria. Types of cultures: pure, mixed, contaminated. Okay. Which media do we use for virus cultivation? We use cells. Because viruses need cells. Cells essentially is the medium. Live organisms. We can use any live organisms as the medium. People grow, yes. So, good. Liquid, mostly indicator or accumulation of a large amounts of microbes. Solid is solid, like agar, is used mostly for isolation, because it gives you like separate colonies. Okay. Live organisms, limitations, cost, for potential possibility of contamination of the organism. You know, animals may acquire their own diseases. Why do we incubate microorganisms in incubator-controlled environments? Main isolation techniques: strict plate, pour plate, spread plate. Am I clear? Why do we need to isolate them anyway? Inspection. Well, you have to say, you know, we can distinguish colonies by appearance, by color, by shape of the edges. We can look at them under the microscope, and so on and so forth. Identification. If I will review it in details, it will be really time consuming. On the quiz, the question number 10, 9 and 10, kind of gives you the heads up on what the question in the exam is going to look like. I'm not asking about intimate details of each method, but I ask basics, okay? PCR detects DNA. Sequencing sequences DNA, not the protein, not the lipid, DNA. Right? Immunofluorescence detects cells using antibodies. Know these basic things. Am I clear? So I'm going to skip this. Okay, genetic identification. Redi and Pasteur experiments. Redi, the meat thing, and Pasteur is the curved neck. Endosymbiosis. Mitochondria and chloroplast is the result of endosymbiosis of the bacteria being acquired by ancestral eukaryotes. Understand the contributions of these four guys. We talked about it. Germ theory of disease. Germs cause disease. What was the impact of each four of them? Each one of them. Main shapes. That's what we started our day today with. I ask you spherical shape, you pick coccus. Am I clear? Cool. Maintenance of the structure. Understand osmosis and tonicity. Okay? Osmosis, movement of water towards the higher solute concentration. Tonicity is the relative osmolality. Cell parts found in any bacterial cell. There are four. Okay, I'll leave it to you. Main components of the bacterial cell. The function of inclusions is. Pick one answer. That's going to be the type of question that you're going to see. I specifically focus on several endospores, transport across the cytoplasm, so no simple facilitated diffusion and active transport. Direction in terms of the gradient and whether they need or do not need energy. Okay. Envelopes, gram-positive, gram-negative cell envelopes. 2 layers in gram-positive, membrane and wall. 3 layers in gram-negative, membrane, wall, outer membrane. Functions of lipopolysaccharide. What peptidoglycan is made of? Well, amino acids and sugars. And that it confers the structural integrity to the cell. Mycolic acid, is the outer waxy layer in mycobacteria? You can answer this question for sure. Difference between archaeal and bacterial. Archaea do not have peptidoglycan. Glycocalyx, capsule, slime layer. That's what we discussed just a few dozens of minutes ago. Difference between flagellifimbre and pili. Function. I always ask about function. Does that make sense? I ask you, which of the following is used for movement? You pick flagellum. Got it? Run and tumble, positive, negative chemotaxis. Okay. This, and then I elaborate on it. Okay, let me just, this is what we essentially discussed in the very last hour of today's class. There's no point in going over each organelle, but it's going to be, this organelle does blah, 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 blah. And you pick one answer with the name of the organelle. Or, the main function of smooth endoplasmic reticulum is, and you have 4 functions, you pick one. Clear? Let me see if there is, yeah. So that's pretty obvious. Now